So I'm going to start with an overview, and this is admittedly an overview some of you may have seen before that I use for a lot of talks and introductions to talks, but I really want to talk about how important it is that we recognize that precision medicine is really dependent on translation. And the better we can translate between the clinic and the bench and the clinical research arena, the closer we'll get to being able to find precise treatments and cures for KCNQ2, epilepsy, and for a whole host of other developmental and epileptic encephalopathies and genetic epilepsies. And I'm mentioning this here now in part because as much as we are not there yet, we want precision treatments, we want cures for all of your children. We're one of the leaders of the pack in KCNQ2. You think about the few genes that we've known about for a long time, KCNQ2 is at the top of that list. So that's great, we've known about it, it's important. It's also one where we have a lot of traction because of the engagement of the community, because of all of the labs and groups who have been working on KCNQ2. And also we've got a lot of traction because it happens that KCNQ2 is one of the most common epileptic encephalopathies, one of the most common genes we see, especially in babies who have new onset unexplained seizures. So 30 years ago, new onset unexplained seizures in a newborn, it fell into that terrible category we called idiopathic, which is doctor speak for we're not sure, we're going to give it a name that sounds like it's something, but it's really we don't know. Fast forward to now, if you were to sequence babies with Ohtahara syndrome, let's say, severe epileptic syndrome with tonic seizures and very abnormal EEG, the majority of those can now be diagnosed with genetic abnormalities. And of those, KCNQ2 is the one we see the most commonly. So it's not the pack that you want to be the leader of. And Sarah, you always say this isn't the club that you necessarily wanted to sign up for, but here we are, and we do have strength in numbers. And so this example and this blueprint that I'm going to illustrate is one where KCNQ2 is sort of pushing the envelope in a lot of ways and is going to really give a blueprint to other genetic disorders.
Can I just interrupt you there for one tiny second? When you talk about being the leader of the pack in numbers and kind of the power of numbers, I'm understanding you to say that it's the most common. Can you just play out that a little bit? How common is KCNQ2 in terms of, these rare genetic epilepsies? How many cases are there?
Yeah, so, I don't think we know, because I think a lot of the kids are still out there not diagnosed. But if we do take a group of patients, a large group of patients, let's say the next 100 babies who came to the neonatal ICU with unexplained seizures, so not from prematurity, not from a hemorrhage, but unexplained presumed genetic epilepsy, and we did exome sequencing, we would probably figure out a cause for at least half of them. Of those 100 babies, half would have an identifiable genetic cause, and they'd be all sorts of different causes. But a good handful of them would be KCNQ2. And I'm being purposefully vague because every hospital is going to decide differently who gets sequenced and will apply resources differently to be able to do those sequencing studies. But it wouldn't be surprising to have a couple of babies in that group have KCNQ2, maybe 2 or 3 or 4 or 5 with KCNQ2 and a couple with SCN2A. These are the kids without the brain malformations that will be another genetic subset. So a good plurality of the increasing number of those we can diagnose. So that's one hospital. Then we take another hospital. So I really think we've not really even seen the tip of the iceberg. So the dozens of patients we now know about are the tip of the iceberg for sure. There are lots of places where this testing is not being done. It's getting better. And it's getting better because of initiatives to provide free testing, but we still have to get out there and really identify patients have, as I'll show you through this little assembly of puzzle pieces I'll put together here, we really have to work towards having trials, but we equally have to work towards having patients lined up to be ready for those trials so we can have the most impact. So that was a long way of not answering your question, Sarah. So
I liked it, and I don't want to interrupt, but you gave me permission to ask questions as we go along. So allow me to let you to continue, please.
Yes. Thank you, but do interrupt. It's more fun for me this way too. So when we see patients in our clinic or in the NICU, we make a diagnosis of epilepsy, recurrent to unprovoked seizures, and we still really have this as our kind of entryway or gateway diagnosis for a lot of the genetic epilepsies, even though we know there are lots of other important symptoms. And that is because epilepsy is often traumatic, and children will show up in the emergency department, or if they're in the hospital and the newborn nursery, they'll end up in the ICU. And so we see patients, we diagnose epilepsy. Nowadays, we do a genetic evaluation more and more. And then we treat our patients, and it's often clinical empiric treatment based on the symptoms. And as we learn more for KCNQ2, we sort of know which drugs do better. Carbamazepine does better. There's a list of drugs that tend to do better based on anecdotal experience, collective anecdotes, but it's still largely empiric therapy. And what I hope to present to you is a series of puzzle pieces that really represent the science side of things that we can then apply into this inner circle of clinical medicine, so that at the end we actually have a whole where there's a reverberation between the science and the medicine. And that all of the medicine really depends on the science, but also informs the science as it's happening. So a key part of that for any defined disorder, whether it's a syndrome like Ohtahara syndrome, or it's a genetic condition like KCNQ2 DEE, is not just seeing one patient at a time, but also seeing the patients as a whole, as a collective, and informing ourselves. And one way we start doing that is through registries and natural history studies. So I'll give you a little bit of detail about what we've been doing in partnership with Dr. Ann Berg from Northwestern and Lurie Children's Hospital after going through this outline. But it's also something that we're doing on an ongoing basis here at Boston Children's, really fueled by your initiatives, your support, both initially with financial support, but also with the enthusiasm and the willingness to give us time. And it's often a lot of time to be able to put data into these studies and to make some sense of it. So that's one piece. Gene discovery, well, as I mentioned, KCNQ2 has been discovered for a long time. We were able to find more and more variants, but what's a really critical next step is expert variant review. This happens in many forms. You'll have heard from Al George, and you'll hear from Ingo Helbig invariably at some point at one of these summits about the many different ways that we can catalog and curate variants. So if somebody comes and says, I've got a child with epilepsy and they've got a variant in KCNQ2, what this means is I'm not signing them up for a trial right away. I'm not even putting them in the natural history study right away, or even putting them in a database where everybody's putting all their information about KCNQ2 right away. I'm taking a step back with Sanal Mahida, our genetic counselor, and saying, is this a variant that's causing a change in the KCNQ2 protein? Is this a pathogenic variant or a likely pathogenic variant? Or is this maybe a population variant, a SNP, what we call a single nucleotide polymorphism that I might have, or anybody might have, that is not affecting the protein? And this is critically important. We're talking about moving towards precision medicine. We're talking about moving toward trials and having trial readiness and having patients ready for trials. And we need the right patients to get to the right trials. So for example, I've seen a number of patients in our epilepsy genetics clinic where a patient may have been referred to our center because we have a trial for thus and so treatment for thus and so gene. And they'll be referred from across town or across country because they want to make sure that their patient, a doctor will say, I want to make sure my patient is going to be eligible for this trial. Can you see? And we'll see the patient, and the family comes saying, hey, we've got this report. It's got this gene on the report. When do we sign up for this trial? And sometimes we say, you know, we're ready. Let's tell you all about the trial. And sometimes we say, you know what, we don't actually think that that gene has anything to do with your condition. And sometimes that's devastating. And sometimes that leads to another whole re-exploration of what might this child have, and leads to a different treatment, and sometimes a precise treatment that leads us to really specific approaches to patients. But it wasn't, you know, they came in door A and they left through door C, but door C might have led to a precision treatment, and that has happened. And so I'm spending a lot of time talking about expert variant review because it's just so critically important. And it's also an area where, again, there's traction with KCNQ2. So through the Center Without Walls and the Channelopathy Research Consortium that Al George is leading, Ingo Helbig, Ed Cooper, a number of our KCNQ2 community friends are working very hard to put information together to curate all of that information. And then they're working doubly hard with a mechanism through ClinGen and ClinVar with Ingo Helbig and Heather Mefford at the helm and Lacey Smith at our institution, looking across epilepsy genes and curating the variants. So today, you have a variant in KCNQ2 come back on a report. Hopefully it's a good report with a lot of information on it about whether it's present in child and parents or only in the child, and whether it's been seen before and various other things. But you can also go to a database called ClinGen and ClinVar and look up the variant and learn more and more about it. As this group is putting in a lot of effort, they've actually got a concerted funded, NIH-funded 3-year effort to bring this sort of information together for KCNQ2 and other genes. So this is really exciting. Cataloging all this information doesn't sound like the most exciting thing to do. It sort of sounds like library science, and it is really, really, really hard. sophisticated library science in a way to put all of the great minds' data and information into a place that's accessible to families and to physicians. So my epilepsy colleagues down the hall can, I still want them to call us and have them let us know when they have patients with KCNQ2 encephalopathy, but they can probably make that call more easily of, is this really KCNQ2 or not, when all of this information is cataloged. So that's a really, really important piece of this puzzle, is understanding that.
And you can probably see me like raising my hand over here to ask a question, because this is a really exciting piece for families. And I'm sure that a lot of families out there are saying, so when could I, go on my computer and be able to find out some of this information? Is there any sort of time frame around that, Anne? What are your thoughts?
A lot of it is already up, and actually we have to thank for that, not academic institutions only, but actually our genetic testing company partners, where they've actually done a phenomenal job of putting cases in. What we haven't yet done, and what we're hoping some of our IT gurus and colleagues will help us do, is to figure out who's who in a way that still respects privacy, but that might actually be able to have an ID linker where you could go and say, let me look up this variant that my child has, and look and say, oh, there's 6 different entries. So there's at least 6 different people with that same variant aren't there. But it could be that I put it in, and Ed Cooper put it in, and the genetic testing company put it in, and a geneticist put it in, and a genetic counselor put it in. So it might be that 5 of those 6 entries are all the same person. And we don't have a way of knowing that yet. So one way of flushing that out is to put a little bit of de-identified but still informative information, male or female. So half are male and half are female. It's not all the same person. So there, there's some simple deduction we can do. But as those databases become richer, we'll be able to hopefully get more information that's meaningful. And eventually what I hope we can do is have some sort of an identifier so somebody could look and say, oh, that's my child. Isn't that great? We're already in there. Or, hey, none of these is my child. What's going on here? Let me call up that neurologist and ask them to push the testing company to push the information in. So that is, it seems like the simplest thing, and has been one of the hardest things to do, is to sort of get a unique identifier that will actually let us connect the dots. But I think that's coming. And it's also critically important to know not only how many people are there, but how many children are there with this variant, and how many are out there with this variant, and how many are out there with this variant, because that is potentially going to be information that's useful as people are strategizing to make some of the very, not just gene specific, but variant specific treatments that people are devising in their labs down the hall from me and Tim Yu's lab and other labs, where, it actually kind of matters what the variant is in some cases. And, if we know that there's 20 kids with this specific variant, that could be enough for a clinical trial, like a real randomized clinical trial that we'd actually have generalizable data to draw from. So these are the things that are really the building blocks that we're resting on. And this whole half of the puzzle is all clinical computer-based research, clinical research, what we call in silico research, because we're not actually using test tubes at this point. But then we switch gears over to the functional assessments. And then we do need, in my lab, FISH, in my colleague's lab downstairs, iPSCs, in our core for translational research here at Boston Children's, our translational neuroscience center, the ability to keep cells in the freezer for long enough to be able to use them for experiments. So we can actually then do functional experiments, move eventually to preclinical trials. Yes, in mice, maybe in other animals, but maybe in other systems where we can check how excitable are these cells, and then move to clinical treatment in the form of precision trials. So we have a sort of precision trial already. Ezogabine was a drug that worked for many children. We've presented that in past summits and actually at an AES, and we're finally kind of putting the data together in a paper that our group is, we've just decided AES abstracts were a good forum, but we want to sort of get it out in the literature so people can find our relatively small experience more easily. But ezogabine is pretty precise in the sense that it's a channel opener, and it actually worked for some of our patients quite well, but then it got removed from the market because it did some things that we didn't like, urinary retention, discoloration of the lips and the retina, which made people worry about vision and so on. That's been a rough go for some families where it really worked really well. And we've actually had, in one case, a family that was able to continue it through means that I won't go into, but they were able to continue a supply and have permission to continue that now. And it's really been sustaining for this child, because that was a precise treatment where we unfortunately had the experience of having to try, well, what if we take the drug off, and saw, wow, the seizures went way up and things were not good, and then put it back on and got a response again. That's not something we want to have to do for every child. We want to be able to have precise treatments that we can have and sustain. And, we'd like to have something maybe more precise than a global potassium channel opener, or a potassium channel opener that'll do lots of other things that are not just addressing our symptoms. And so lots to do on this left side of the pieces of the puzzle. And I'll just give you a little hint into one of the experimental plans we have as well.
So I wanted to go back 1 second, just one quick thing. You can stay on the slide, but when you talked about the natural history study, it did occur to me that that's an opportunity for a shout out for families. How important is it, in your view, for families to register, to take part in this natural history study?
So super important. I'll give you some results of what we found here, but each of these little dots, and I'm sorry that they're little, there's very little dots, but each of these dots represents a child. And so they're little dots, but it's a big deal. And I've called this slide Many Patients, One Protein, because this whole community is converging on KCNQ2, but each of these dots, each of these variants in these various different parts of the KCNQ2 protein, the voltage sensing part of the protein, which is really important, the C-terminal part of the protein, all of these different areas are very important. And this understanding that we have goes back years to Ed Cooper's work and others' work, where we have begun to understand, what does this channel do? What is it functioning? What is its function in the cells and in the developing brain? But each dot represents someone who signed up for the natural history study. And so, yes, we want natural history, but those families who enrolled, many of them, when we had a summit locally here in Peabody, and people went to great lengths to not only enroll, but get blood drawn and do all sorts of things that took a lot longer than they should have. But we sort of got the village together and were able to make that happen. That informs our understanding of the spectrum of KCNQ to DEE, not just from what symptoms do you have, but what's going on with this channel. If we have a drug in the future, let's say, that's going to really help with people who have an abnormality in the voltage sensor, that's good, but then we've got to know that your child has a variant that's affecting the voltage sensor. And so this is really a piece of the puzzle that builds upon natural history study enrollment. Anybody who's in any study related to KCNQ2 really needs to have the variant mapped on some structure so we can understand the context. This also helps us strategize when we've got lots of patients enrolled and we're into the 70s plus at this stage. We not only can make a cartoon like this that says, okay, we know what we're talking about with the different parts of the gene. It can also say, well, if we've got limited resources to do things like iPSC studies, and they're very, very person and time intensive and resource intensive, let's make sure that we sample this protein. Let's make sure that we look at a variant or two from the voltage sensor, because we know that's really important. But let's look at this intracellular N-terminal region as well, and let's look at the C-terminal region as well, and let's make sure we've sampled the regions where there are patients. And so, sorry, my lights just turned off. Not moving enough in here. But let's make sure.
Do you want to take a minute to get that?
I'll just move over and see if I can get the lights to turn back on.
No worries whatsoever. There you go. Absolutely.
There we are.
Yeah,
so now
I mean, now you're back, and I'm just struck by the fact that that point of one dot is one person. I mean, the power of what you've been able to do with participation. Please do continue. This is an exciting slide to see.
Yeah, so we put this together, myself and Sanal Mahida, joining forces with Ann Berg, who called us and said, I've been talking to families and I've been looking at data and parent reported data, family reported data, and I'm seeing some really interesting things. Do you want to hear about it? I said, yes, because we're seeing some interesting things as well. And so we put our notes together, but we were able to then organize the whole story around this protein and say, well, let's look across and make sure we've covered patients who are in all parts of the protein so we can really sort of understand this better. And then eventually look and say, well, are there differences in symptoms if you've got a voltage sensor variant or a poor variant, which we haven't really gotten yet. Maybe that will come, and maybe different subtypes of electrical abnormalities that are sort of at the level that Ed Cooper and Al George will be able to do with their cellular studies, that may help us to distinguish as well. But again, why do we care? Why do we want to do that? It is cool science and we like that. But it also might be important because, again, when there's a trial in the future, it might be that if you've got this kind of an electrical disturbance and a poor variant, you could be in this arm of a trial versus another. And so I go back to this diagram. We started making these diagrams when we started enrolling patients. And I think they're vitally important as sort of a backdrop for all of our studies, whether they're clinical research or they're bench research. So I'll tell you what we found. And this is, again, going back to this idea that we need to have natural history. We need to really catalog what's going on with patients and variants. And I need my head to stop blocking that part of the slide. So there we go. So this is what you've told us. And I know many of you are in the audience and you've been the participants in studies, and this is what you've told us. And some of this might not be a surprise, and certainly isn't a surprise to neurologists, but I think it's important to look again at the numbers. We had a number of patients, and we included patients who had for sure variants in KCNQ2 and variants that we could vet and say were pathogenic or likely pathogenic. And what we did was we found that a lot of patients had bilateral tonic-clonic seizures at some point in the stage. And when did they start having seizures? A whole range of different ages. And I highlight this because in my mind, when I was a trainee and I heard about KCNQ2, it was the benign infantile presentation with tonic seizures. And as Sarah Veckhusen showed us with her group, when they looked at KCNQ2 and KCNQ3 in babies who had tonic seizures and encephalopathy, KCNQ2 was really sort of founded as an encephalopathy gene in that era. And so even now, even though we've been through this for so long and we've seen all these families and the wide range of presentations, my synapses go right to tonic seizures, as they should, because it's a big part. A third of the kids have bilateral tonic seizures, but look at all these other seizure types. And so here I am as an epileptologist. I've been doing this for a while, but I looked at the data with Anne. I said, wow, this is actually not necessarily what I might have expected. Now, there are hemiconvulsions in one child, not surprising to me, because Ingrid Scheffer and I put together a whole group of patients with epilepsy of infancy with migrating focal seizures, a mouthful subtype of severe epilepsy. And what gene was there first and foremost? That was KCNT1. And what gene was on the list of, hey, we see a few of these as well? KCNQ2. And so, it's not just the Ohtahara syndrome, it's not just the tonic seizures, it's all of these different types of seizures, which we know. And some of these subtypes of seizures, particularly the myoclonic and the infantile spasms, they may be a little bit different, and their biochemistry and their electrophysiology may be a little bit different as well. And that's what I think the next couple of years, hopefully putting all of those pieces together, will help us to probe. But what you told us is lots of different seizure types. And then what you told us, you've always been telling us this, but what Anne was really able to demonstrate really nicely was that seizures are really only one part of the story. We have this hypothesis with epileptic encephalopathy, and we have this hope as neurologists, I'll say, that if we could treat the seizures, if we could just treat the seizures, everything else would get better. The electrical disturbance would calm down and the development would be set back on its correct path. The behavioral disturbances wouldn't be so bad, or they would correct themselves. And we know, and we've known from the beginning of the KCNQ2 story, that for KCNQ2 in particular, that's unfortunately not always the case, and more often than not the case, because the seizures will often subside. Sarah reported that with her first paper with KCNQ2 encephalopathy, and many studies have now reported this, and Anne really got that straight from the parents, that it doesn't matter if your most recent seizure was a few months ago or a couple of years ago, there's still a lot of impairment. And so Anne looked at the number of domains that were impaired, and this is motor, language, social, being able to take care of themselves. And it didn't matter whether your seizures had been treated fully for a long time or if you were still having ongoing seizures. The level of impairment was still rather serious and profound in many cases. And the number of seizures you had recently and how long ago was your last seizure actually didn't matter so much. And just to give a little bit more of a description and to put some of the words in front of you in terms of what types of things were going on, this was looking at independence, looking at communication, and looking at the ability to feed independently. And these p-values, for those of you who follow statistics, are not significant, meaning there is not a difference between the different groups of the kids who had their most recent seizure less than 6 months ago, up to 2 years ago, or more than 2 years ago. So the colors look a little bit different, but it's actually not a significant difference. And so what this means is whether your seizures went away a couple of years ago or more, or even more recently, the same number of children, the same relative proportion of children, were still completely dependent on caregivers and parents, partially dependent, and only very few of them were independent with no constraints. And then if we look at communication, likewise, we've got children who rarely communicate, who are inconsistently able to communicate with familiar individuals, effectively communicate with familiar individuals, and then onward down to effective with familiar and unfamiliar, which was, again, unfortunately a rarity in this group. And that didn't matter. So even if we got your seizures to go away with medications, or even if they went away on their own, this degree of impairment of communication, and then also with ability or inability to feed independently, didn't matter. So, not what the epileptology community necessarily wants to hear, because it sort of makes you take a big step back and say, these are the things that are important. These are the features that are really important to the patients, the families, and need to be important to us. We need to be doing a better job of cataloging these features, because what are we hoping for? We're hoping to make that circle full and go to precision treatment and have trials, and we need to know what we're looking for in the trial. So, this, on the face of it, might seem like a fairly straightforward, simple, descriptive set of data that Anne cataloged, but she cataloged it so beautifully, and she made categories that were really meaningful based on input from families, and we're able to demonstrate that, yes, of course, we want to limit seizures if we can, but that is only a small piece of the story for KCNQ2. And again, this is not news to most of you, but it's something that we have to sort of demonstrate, because when we go to our partners in pharma, and we go to our partners at the FDA, and we go to our partners in clinical practice and say, hey, do you have patients who might be interested in a trial? We're not necessarily talking about a seizure trial or only a seizure trial. We're talking about other things and other designs that have to be thought about.
And that can be, just to jump in at this point, this is an example of how important and powerful your research and studies like Anne's are, because it isn't, it's those co-expressions, to use Anne Berg's lovely word, I think far better than comorbidities, those co-expressions of how KCNQ2 affects an individual, which are so important. And I think what I hear you saying is that they will be relayed to pharma, to companies that are looking, that they will be included. Do I have that right? They will be included as scientists and pharma look for treatments. We'll be thinking about those broader ways in which KCNQ2 affects individuals. Is that correct?
Yeah, and it has to be. I mean, if we do a trial for kids 6 and over with KCNQ2 and want to look at seizure outcomes, well, we're not going to have a lot of people there to enroll, right? I mean, that may be an infancy trial. Could we look at seizure reduction, seizure freedom in infants and see how that then correlates with long-term developmental outcomes? Yes, we'd love to do that. That's why we need early precision diagnosis to find those infants and offer those trials, and start with the safest ones we possibly can, and then move on to potentially more high-risk treatments that we might be talking about in the future. But that's the pipe dream, right? Let's get people at diagnosis, and let's try to treat right away, or better yet, let's find out before a seizure start and try to prevent. Our colleagues in the TSC community, the tuberous sclerosis complex community, they have a different sort of a bellwether symptom. They often have cardiac lesions. So these babies are diagnosed because of heart lesions as having tuberous sclerosis complex, and they can be followed, and EEGs can be done at the first turn of events that there's an electrical abnormality. They can be put on vigabatrin to see if seizures can be prevented. And we know that upwards of 90% of them are bound to have seizures. We know that from good natural history studies. And so we can then say, well, let's see if we put these babies on vigabatrin, the high risk ones, let's see how many end up with epilepsy and how many might be spared the epilepsy and the encephalopathy. So that's a really cool design, and that's a really amazing opportunity to do that. I'm not saying we want heart lesions, we don't want heart lesions, but we don't have that kind of a non-seizure, non-developmental bellwether symptom for KCNQ2 and for most of our genetic epilepsy. So we're really often saying, let's find you at first symptom, jump on the diagnosis and make it, and then we can jump in with trials. And that's something we have to do as a collective, as a community, through groups like the Pediatric Epilepsy Research Consortium and other large groups of individuals who can get together and participate in trials together.
Yeah, so, that makes a ton of sense. I'll allow you to continue.
Well, so I'm going to just do one little brief glimpse into the science experiments, because those are sort of, material for whole talks, but I just, again, want to emphasize how important the whole puzzle is and how these registries and natural history studies can also drive the science. As I mentioned, it tells us which areas of the protein to focus on. It also has us as physician scientists in front of real people, real patients who have real variants that can be vetted, that then require further study and can really serve as a platform for further drug discovery. So what are we looking for in our models, whether it's cells or mice or whatever it may be? We're looking for sufficient genetic homology to humans. So if we model something in a mouse versus in a fruit fly, turns out the mouse has more genetic homology to humans. We still need to know that a protein like KCNQ2 is expressed in the mouse, but the higher up you go, the more homology you get. It tends to be a better model, but you can't always do as many mice and as many different variants as you might want to. So you do a few strategically. Maybe you take one from the poor region and you take one variant from another region and you make those models. So that's been done in mouse. Amy Brooks-Kale, one of my dear colleagues from over the years, has this wonderful expression that we need models because the perfect model would be a human, but there are things that we need to do in models that we would never do in humans. And that's why we need these models, whether they're animal or cellular models. But we can sort of compromise between let's do animal model studies and we need as close to human as possible by taking human material. So when patients signed up for our registry and also gave samples, we said they're going to be in the freezer till we have the funds to be able to do some iPSC studies. And we're certainly not the only ones doing this. Al George's group is doing iPSC studies on KCNQ2 and has published some nice data already. And so we've been able to do this, and this lets us take a human cell, blood cells, and zap them into pluripotent cells and then turn them into different types of neurons. And so we've been able to look at cortical neurons in that way. So that's something. I'll just give you a little bit of our roadmap as we're going through that. But that's a really important thing, because it would be great to study anything in any model that's there and genetically tractable, but we would need it to be a model where it actually has relevance to us, because we want this to go towards clinical trials. So genetic tractability, can we make variants? Can we mutate these models? We absolutely can. And more important, we can correct the variants. And so if we've got a cell line with a pathogenic variant in KCNQ2, we've got really readily available tools to be able to correct it in the dish, to fix the variant and see if that fixes the abnormality that we're seeing. And then what are we looking for? We want to look for anything that's meaningful. And from a neuroscience perspective, we want to look at anything that tells us about how the brain develops, anything that tells us about how KCNQ2 functions, what does it bind to? Well, we know it binds to KCNQ3. What's that complex doing? What's the stoichiometry and how many units of 2 versus 3 do you have? And that's really important and foundational and fascinating. But from a translational perspective, we would like to see some kind of seizure-like phenomena or electrophysiological abnormalities, some evidence of hyperexcitability, because what do we want our drugs to do in the dish or in the animal? We want to dampen that hyperexcitability, make these animals and cells look like non-affected animals and cells. Now, if we really want to get sophisticated, and if we have animal models where we can test these things, we'd like to look at learning or behavior abnormalities. We'd like to look at things like sleep abnormalities, which we can increasingly do in zebrafish, in mice, and other model systems. So listening to the families, hearing what are the symptoms that are really at the core of what's going on, not just on the EEG and what we look at in the clinic sometimes, but what's going on day to day that we really would like to see corrected, that we know is due to KCNQ2 dysfunction. So these are the types of things we'd like to model. And then for drug screening studies, you'd like to choose some models where you can get the drug into the models easily, and where you can do a lot of experiments at a time. So this is where cells and small animals have an advantage over the large animals, because you can do 96 well plates full of cells or 96 well plates full of fish or other small creatures, where you can actually do a lot of experiments all at once. Once you have some traction there, then maybe you go to the bigger models and look for a little bit more proof of principle that a genetically homologous animal is going to show us something. So these are general principles, but they absolutely apply to us in the KCNQ2. And so this is a case that I've talked about before, but this is a patient I've seen for some time. And we were fortunate to have not only cell lines, but be able to take those cells, make them into initially motor neurons. Why motor neurons? It's sort of an interesting coincidental backstory that the ALS community is really interested in KCNQ2, and retigabine is what we call ezogabine, and have had that. And so we first started with motor neurons because we could, because there was a lab downstairs from me that was studying motor neurons for ALS, and they were willing to take on KCNQ2. We've continued that collaboration and that conversation, the experiments now for years. We had a big pause during COVID, but we're kind of back in action now. And now we're making cortical neurons, the type we see in brain cells, and we're seeing all sorts of firing. We also had some interesting data come up at a poster at AES where Maurizio Tagliatella and Sarah Vecchus and I sort of had this moment of, wait, is it gain of function or is it loss of function? What does that even mean? We're seeing evidence here of this and evidence here of that. It turns out that as the story continues, we continue to be equally perplexed, because reproducible, robust data from 2 different labs looking at 2 different measures are showing us different things. And so maybe what this tells us is we have to be careful about how we characterize these things, and the gain of function and the loss of function is a little bit of a through the lens that you're looking at. At the end of the day, what we are seeing is hyperexcitability, which gets me back to what I mentioned on the previous slide, is the core phenotype that I'd like to see is hyperexcitability that we could then screen for and dampen with drug screens, for example. So that's sort of how we walked into this whole team of individuals. We described the variant with Sarah. We had the clinical correlation with our nurse practitioner on and off ezogabine. We actually then did these experiments in the motor neurons, and then went on now with a whole team of people looking at the cortical neurons. And Maurizio, whose name I'm hiding, sorry, Maurizio, is doing some ongoing experiments. And then we're hoping, this has been a real labor of love from Maurizio's side, where he's been bringing this whole team of people together. We're hoping to report this as the conundrum that it was, and kind of tell the story about how this wasn't a straightforward do a sequencing change assessment, vet the variant through bioinformatics, okay, we're done. This has been something that made us all scratch our heads and say, wait, this isn't exactly what we expected. And for all sorts of reasons that we've talked about before, and we can talk about in more depth another time, but this made us really wonder, is precision medicine, is precision variant assessment going to lead to individual variant specific treatment? Are we going to need to go to that depth and that level? And I'll say, I hope not, because we don't have enough labs to do that sort of drug development. I hope we can do classes of variants and classes of patients, and at least get people organized by where in the protein are you with your variant. But we'll see. And this one variant from my one patient, to whose family I'm so grateful, is informing us a lot about what we can do, but also letting us know about what we don't know. It's keeping us very humble. So that's an important story and a message I'd like to get out there, that we've got a lot of work to do. But each variant, each family, each person who gives us their data is informing us in a different way. So with that, I'd like to leave you a little bit of hope about where we're heading. Precision diagnosis does require basic neuroscience, and thankfully we've had a lot of that going on in KCNQ2. So we're very lucky. We've had gene discovery, molecular studies, mice being made, lots of ways that we're understanding at the basic level what KCNQ2 does. And that's here in the US, it's over in Australia, lots of different labs in Europe working on this together. So that's amazing. And as I mentioned, it requires variant curation. We've got the tools in place, the people on board, the experts in place. The expert panel that's curating KCNQ2 is your own group of Ed Cooper and colleagues who launched that and said, yes, we'll be part of this effort, but why don't we just take on KCNQ2, because we can do that, and they are. And it's an incredible tip of the hat that was one of the many genes leveraged to be able to have this broader umbrella for genetic epilepsies get support from the NIH to be curated. So thank you, KCNQ2. And then where are we going with precision treatment? And I know this is sort of everybody's goal. It's my goal, and it's where we all want to be. And I'll say, we need those preclinical studies. We need preclinical data about specific mechanisms and drugs, which is going to help us figure out which patients might be amenable to treatment with which drugs and strategies. And then we have to do this whole back around that puzzle piece back to the clinical research side again, to the science, the practice, and the ethics of clinical trials. So what are the meaningful outcomes? That's not just, hey, what do you think, Sarah? What should we do? That's a science. And Anne Berg has really shown us how to do this scientifically and how to look at this in a really robust scientific way at what are going to be the meaningful outcomes. And can we do a longitudinal prospective natural history study at some stage? I'd love to. I think some of the companies who are in drug development for KCNQ2, just as they have for some of the other genes, might be amenable to saying, let's enroll 10 people from each site where you can enroll patients and follow you longitudinally. So when drug X is ready in 1, 2 to 5 years, whenever that is, then we'll have each person's own timeline for seizures, for development, for behavior, all these scores and scales that Anne has shown us, Anne Berg has shown us we can use in this group. And then let's go, let's put on the drug and see how symptoms change or don't change. What are we going to do in terms of inclusion and exclusion criteria? Who should be included? How old? How is there a too young because of safety concerns? Is there a too old because the seizures are gone? I think we've answered that latter question. The seizures are going to be gone for so many kids, but there's other symptoms, there are other conditions, really shouldn't be considered their other conditions. Those are the KCNQ2 manifestations, and those are going to be needing treatment, likely more than the seizures for many of our patients who are older than the infants. And so is there a too old? Probably not. I mean, this is a unique situation with pediatrics and rare disease because we're often sort of waiting for the adults to have the trials and then we trickle down into us. This is going to be one where the kids are going to lead, and there's going to be, I think, substantial push from us to sort of have an infant onset trial and trials, but also to have later onset trials. And we've got to figure that out. You know, that might be by consensus, that might be by getting potential trial PIs together saying, would you be willing to offer this to this age group, this age group? And then I mentioned here stopping criteria. You know, if we have a trial that's going to set out to treat developmental trajectory, you've got to keep that drug or treatment on board for a while to see if there's a change in developmental trajectory. That's not a 2-week trial or a 2-month trial. That might be a 6 or 12-month trial at minimum to see if there's a meaningful improvement. How many cycles of that is a family going to be willing to go through before they say, you know what, we've been on this drug, we have to go to this center, we have to have all these assessments, we've got to do X, Y, or Z to maintain this treatment. How long should we do this before we say yes or no, we have or haven't had an improvement? If we've had an improvement, no one's going to ask, right? We're going to just keep going. Let's go. If we're not sure and we do some objective measures and we don't think there's been an improvement, maybe we should find another strategy that might be better and not pursue a course that's not helping and that's taxing the family. And so that's really important. It's something that we sometimes forget about in our clinical practice. Hey, let's try this. It's not working. Let's add another drug. It's not working. Let's add another drug. And we sometimes forget to stop until the families will come and say, you know, is it maybe possible that we could be not symptom-free on 4 drugs instead of 8? You know, could we give that a try? And, you know, this is all the more important for a trial, a clinical trial, because we really need to figure out how long are we going to try this? How long are we going to subject the children and their families to the trial experience, which is on the one hand hopeful, on the other hand not easy. So that's a whole set of discussions that has to happen. And then whole lecture for another day, a whole discussion that I hope you'll all be having. N of 1 trials, single patient, single variant versus N of few trials, single variant, but a few patients with the same variant, or single gene trial, but really complex strategy like an antisense oligonucleotide strategy where we probably can't do this to start with 50 patients, even if it's a single gene and anybody in the gene would count. We've got to figure out, how do we choose who's going to be the first and who's going to be the next and who's going to be the next. And that's where these criteria, objective criteria, have to be worked out in advance. And again, where my cartoon in my head of this protein comes into play. If we've got a strategy that we think is going to work really well for patients who have variants affecting that pore, let's do the trial for patients with variants affecting that pore and learn from it, and then see if we can broaden that out to patients with variants in other regions. But it's got to be objective. And so that's where I'll leave you. I mean, I'll leave you with a final note of not to sort of remove the enthusiasm, because I have a lot of enthusiasm and hope, but we've got cautionary tales from a lot of our sister genes where everybody thought this was going to be a great idea. Everyone thought KCNT1 was going to be dampened down with quinidine, and it was going to be, the answer to everything, because in the dish, that's what happened. And in a first child, that's what happened. But in subsequent kids, it didn't necessarily help so much. And adults with KCNT1, it really didn't help as much as it should have. And it really caused a lot of bad heart arrhythmia. So we've got some cautionary tales, even in precision medicine and epilepsy. We've just got to be mindful that we want to have progress, but we really have to not do things that we really wouldn't all buy into together and that are going to potentially cause harm. And so it's a lot of work, and it's a conversation. And I think I leave you with the idea that this is a conversation among all of us, the physicians, the scientists, the physician scientists, and you, the patients and families, about where do we all fall on these different parts that are somewhat due to, they're really due for a conversation and need for consensus opinion.
So, it's fascinating. Everything that you said is fascinating. Two things that I want to mention just now. One is that as I am listening to you speak, and we've had you speak at previous conferences, we've talked about lots of things, and I want to come back to that beautiful thank you slide with your team showcase there. But just quickly, I'm conscious of how much things have changed, even over the course of the last few years, by virtue of all that you've learned. And for our families who are joining us, who have been with us from the beginning, and the families who are newly diagnosed, I just really want to make that point, because we don't only understand it better, but we understand it differently. It's more complicated maybe than we thought. So that's one observation that I have. Is that accurate? Does that square with what you would say?
Absolutely.
All right. And the second thing I want to say is, you mentioned something that may not be familiar to our families, particularly the newer families, but maybe some of the others who've been with us for a while. And that is a protocol known as an N of 1 trial. And that's made a lot of headlines. There is an N of 1 regarding KCNQ2 as well, and it's there at Boston Children's. So I just wondered if you could just briefly speak about what you mean by an N of 1. And kind of set that in the broader context of more traditional clinical trials.
Absolutely. And, I should say that this is really all reflective of work my colleague and friend down the hall, literally down the hall, Tim Yu, has been working on. Really, really clever strategies in the dish to try to target a gene and target a variant. And basically, to put it very simply, swap out the area where there's a variant and swap back in some normal KCNQ2. Sounds simple, it's pretty complicated. And you've got to know all of the genetics of the gene and everything around the gene and some surround DNA. And this is only possible because of the genomic knowledge we've gained in the last 10 years, very broadly speaking, with Tim's lab applying it very specifically on a couple of genes. So there is a KCNQ2 program in terms of trying to understand how amenable the gene is and the region around the gene is to these sorts of strategies. There isn't yet an N of 1 trial. There's sort of a desire for a trial, and there's a program in place to try to move that forward. And that's really based on the infrastructure that Tim's team set up to start this N of 1, meaning treating a single child. And in the first trial he did, the trial in quotes, because it's a single experience. The first was a single child with something called Batten disease, with a specific 2 variants that were unique to that child, will probably never occur in combination again. And it's a devastating disease with a downhill course where, you get back to my do no harm, the harm's been done by the disease already by the time this child is diagnosed. And, we really we're hoping to maybe slow down progression of disease, which, and from a seizure perspective, immediately, yes, there was a reduction, but overall the disease continued. And is that because we were too late? Is that because it just didn't work in the way we wanted it to? Hard to say. And so this brings up some of the challenges when it's a single case, a single person. It's very hard to generalize. But we learned a lot about the process, the scientific process, the importance of precision diagnosis, of looking at the genome when you don't have an exome. If a baby comes with tonic seizures in infancy and has a burst suppression pattern, and we think it's Ohtahara syndrome, and we do an exome, we don't find KCNQ2, somebody better do a genome and look for KCNQ2, because that's KCNQ2 until proven otherwise. And we should look around the gene and for rearrangements around the gene. And that's the kind of genomics that Tim brought to that first case, the Mealison case with a single patient. We've got to do that now for every gene. So that was instructive. Get it right, get the diagnosis right. If it looks like KCNQ2, look as hard as you possibly can for KCNQ2. So that's one lesson. And that's a lesson that when the write-up of that first Mealison case was being drafted, I got to put on my co-author hat and say, you know what, you're underselling that part. That's the reason you even got to do the next part, is that you figured that out. That's really key. So that's part one, precision diagnosis, which is always an N of 1, because it's that child and their diagnosis. Then the whole platform. How do you go from knowing the gene, knowing the variant, doing some experiments in addition, then getting it to the point that the FDA says, okay, it's all right to inject that into a child's spinal fluid? That's a lot of steps, and a lot of people, and a lot of teams, and a lot of institutional support and backing from a hospital like ours, and also Denver with Colorado Children's being willing to accept the treatment once it was made and continue it there, which is where the family was from. So quite a lot of bureaucracy, quite a lot of regulation and steps to go through. And I mentioned this to say that these are all really, really, really important, the regulatory committees, the oversight committees, because Sarah, I agree with you 100% that things have changed so much, but I will say that the things that haven't changed are the basic principles of what we're talking about. In fact, I borrowed some from some of my old slides. I borrowed some of those things because it was like, I looked back at them like, you know what?
It's great.
It's that's stuff, the same basic principles. It's just, we've filled it in with so much richness of information, and we've now got new technologies, but it's the same roadmap. It's the same, but we've just got a richness to it that we can now lean on and branch out from. So that's what, at the N of 1 experience is tantalizing, and people every week in my clinic will come and say, all right, so now you figured it out. This is great. It's been 2 months, 2 years, 2 decades, which I'm not really thrilled about.
That's what it probably feels like.
Well, got it, where's
it's
mine?
Amazing. Exactly. And that's going to be a challenge. But I really do take your point that it's like, on the one hand, it's very different. On the other, we're dealing with some essential truths, if you will. And I want to point out for families, and we've spoken about this elsewhere in the summit, but this is an exciting time. And you had such interest in KCNQ2 that you've just, if you could just briefly in a minute or two, explain what you've done with Cambridge University Press, also with Al George, also with Sarah Vekusen, but just that notion of, studying a gene and really having that out there for the community. I've had the opportunity to work with you on it. It's a very exciting project. Can you tell us a little bit about that as we wrap up?
Absolutely. So we were asked, a few of us in the epilepsy genetics community, if we would be willing to edit a series called The Elements. There's a Cambridge Elements series, and this is admittedly not often a biomedical topic area that they choose, but they decided they were willing to branch out and take a chance on us. And so myself, Dan Lowenstein, Aaron Heinzen, and Al George sort of got together, laid out some possible topics. They sort of accepted our proposal to do this. And one of the things that we thought we would do, that COVID unfortunately got in the way of, but one of the things we thought we would do is roll this out a little bit with Al's guidance, with KCNQ2 as one of the first genes that we would highlight, in part because it's common. I mentioned why it's important and why everybody should care about it, but also because we've got Al, who's running this Center Without Walls, and we have Sarah, our friend, who has a whole life as a broadcast journalist and can speak really well in front of cameras and can actually get enthusiasm from people and draw people out. And so we asked Sarah to be involved. So I'm going to put a little light back on you, Sarah, but to be involved as an associate editor, because we didn't want to just write more reviews, because we're all on our computers all the time. Now we're in Zoom all the time, and we're writing and we're doing reviews, and they get put in PubMed, and they're great teaching tools and they're great learning devices for the people who write them. And it's wonderful that we have that medium, but they're not jumping out on the page. And we wanted to provide material from families, physicians, scientists, trialists, in a unified fashion, focusing on the genes just the way you do in the summits, and sort of bring to life a little bit of the energy that we have when we meet in person and you get these groups of people together. And so, Dan achieved that really well at a Half Moon Bay Precision Medicine and Epilepsy meeting in person several years ago. And I kind of wanted to capture that a little bit, that let's, say you scientist, you bench person, you clinical researcher, you family member, foundation leader, can you get together and can you summarize for all of the above categories of people, but for students and for the nurses who might be taking care of your kids, for the EEG tech, for the person who's thinking about doing a PhD thesis in neuroscience and might want to do something on epilepsy. What do I need to know about KCNQ2? What's important? And to not just write the story, but tell the story. And so we weaved in interviews, and Sarah's going to be giving some of those interviews, and we'll be airing some of those interviews through the first content element. We've got an intro element up, but we've got the first content element that's close to actually ready for production with Sarah and Al. And what we realized with our first content element being an introduction is that the two-dimensional web and the interface that we had, it didn't bring it to life enough. And so we're actually now going to beta test a whole new set of systems. So when people go to the KCNQ2 element, they're going to see Sarah and be able to click on a video rather than have to dig around for it. And it's a work in progress, and KCNQ2 is going to be our first content one. But my hope was that it is something we can update every couple of years. So it would sort of be a nice way to have a synopsis. But in some ways it would give us a platform. So when we leave the summits, all excited that we've been able to talk amongst ourselves and really, start new projects and new discussions, that we'll have a place to reflect on that and have it be there, hopefully in PubMed in a couple of years as well, but to have it be there so that maybe people who couldn't attend the conference will be able to look to. But it can also sort of be our report card. How did we do? It's been 2 years. What did we write last time? We didn't really do as much as we said we'd do here. We got to get on this. And in a little bit, it'll sort of help us to benchmark a bit, I hope, is what this platform can do.
Well, it's super exciting. I'm so thrilled. And I know many people in our KCNQ2 community have already begun. You participated in so many ways. So big shout out to the KCNQ2 Cure community as well for assisting with that, because I think it will go back to help us as well. And it has been phenomenal to have this hour to chat with you. Thank you so much for the gift of your time. We know how busy you are and how many things that you're doing. And we want to thank your team as well. We see some pictures there on that slide, and we want to send out a really heartfelt thanks to all of them, particularly because you're all medical professionals. You're all there working through COVID, in your PPE, being part of, keeping us all safe, but also continuing your research in the lab. So a huge thank you from all of us, and for that fantastic presentation.
Thank you so much, Sarah. And, we'll be talking about ways to engage and to enroll and so on. And I just wanted to give a face to the name Sanal Mahida has been working with us closely as a genetic counselor, vetting all those variants and curating, and our wonderful research assistant team, Brandon and Julia, have been on the other end of the email that we have and the phone line. And Brandon in particular has become our KCNQ2 guy. And so if you do enroll and you hear somebody on the phone or you Zoom, it's going to be Brandon. Happens to also double as a jazz musician with Phil Pearl on the side. So maybe one day we'll be able to have you experience that live.
We're down to that. That's great.
But yeah, these are the
anyone else you want to mention on the slide, just so that we can see your team there?
The whole team gathers every year at the, mostly we had to skip a year with COVID, but we try to gather at our homes, and we gathered this year, thankfully, during that little pause when things were looking good, at Beth Scheidley's home. So Beth Scheidley is our co-director of the Epilepsy Genetics Program, and has this wonderful knack of bringing out an ice cream truck. So we get the whole gang and families and so on, because this is really it's a village and a family that comes together. And, everybody knows about KCNQ2. Everybody's looking for KCNQ2. And some of our team members now are doing a project where we're doing genome sequencing in the NICU, and we're doing this in partnership with Australia, with Royal Children's, and also Great Ormond Street in England, and Toronto Sick Kids up the road from us. And one of the justifications we made to our institutions to do genome sequencing in the NICU was, if we don't do this, we're going to miss KCNQ2. So our whole team hears about it all the time. And so even though not everybody's focusing on this gene, they're hearing about it and they're aware of it and they're looking for it. And so this is the team that's behind all of the research.
Well, thank you. Thank you again so much. We really appreciate it. And we look forward beyond belief to seeing you and as many of your team as can join us in a year when we do this again in person. And we look forward to that too. Thanks so much, Anne.
Thank you so much, Sarah.